spread the word. Visit our website for some updates. And thank you.
I'm a medical doctor by training, so I don't know anything about data science, which puts me in a very nice position because I can say a lot of nonsense and I can ask Serge to do a lot of difficult things. You can always say, well, why isn't it possible? So when he has a tough job, actually changing my questions and opinions. So as I said, I'm a medical doctor. I've worked for more than 23 years now in the pharmaceutical industry. Started at Janssen, then left to work with a biotech company, which was acquired by Johnson & Johnson, so we came back to Janssen. And in the meantime, we developed, together with Marie-Pierre, who is here as well, 3 drugs for HIV, which are all 3 on the market. And the first 2 were specifically aimed at treating HIV patients whose virus had already seen a lot of other, or at least one other therapy, and had mutated so that the therapy was no longer effective. And at that time, there were patients who basically didn't have much or many options anymore. And although HIV was treatable in the sense that you can basically turn it into a chronic disease now, you can do that with good medication that shuts down to a large extent the replication of the virus, and therefore there is no further immune damage, and you don't have all the nasty diseases that occur in patients who are not treated with HIV when they lose their immune system, basically. So you can do that very well, but if the virus, and the virus is notorious to be able to mutate around the drugs, then you need other drugs, more potent drugs. And we worked on 2 of those, and I can say they've made a big difference in the life of many patients. We also had a drug that is actually aimed at first use, so what we call first-line treatment in HIV. And after those antiretrovirals, so drugs for HIV, the group developed 2 drugs for hepatitis C, and then the last one we developed was a drug for multi-drug resistant tuberculosis. So you have a similar situation that many patients in the world, their tuberculosis has become resistant to most of the drugs that exist. If you have drug-sensitive, what we call tuberculosis, with 6-month therapy and 4 drugs basically in a regimen that lasts 6 months, your chances to be cured, if you have access to the medication and you take the medication, your chances to be cured are pretty high. But for some reason, if you don't have access to good drugs, or the TB is not treated well, then again, similar as with HIV, you get resistance, you need other drugs. And we developed one, the first novel drug in 40 years, and it makes a huge difference in treatment of drug-resistant TB. Why I'm giving all of this, I'm not bragging about this, but why I'm giving you this information is to come to the global public health part. You can think probably, why is a pharmaceutical for-profit company interested in public health? What is behind that? Are they trying to find money where others don't find it? Well, I can tell you there is no money to be found. That is not a motivation. The motivation actually comes from finding drugs that make a huge difference in the life expectation and quality of life of patients wherever they are in the world. And like TB, and especially multi-drug resistant TB, is not very frequently found in the developed world. It's a disease of the poor. It's a disease of those who don't have access to drugs, cannot afford them, or the healthcare system doesn't afford them or doesn't pay for them. So in many parts of the world where patients actually don't have access because they cannot afford it, that's where our organization, because we had these innovative solutions, were
focusing at. And it's fantastic to be able to do that in a company. And it's not like we have a blank check and we can go to the J&J bank every day to get money for this. We have to be very, we have to make sure that we deal with the resources that we get in a very efficient way. And we're also trying to leverage our own internal investments by external funding. So we do all of that in partnerships. But it is aimed at innovation for resource-limited setting, unmet medical need, neglected diseases, or better neglected patients. Because there are many diseases that have already a cure in the developed world, but patients in the developing world don't have access. So for them, they're also neglected. So that's what we're doing in the organization. We do it for TB. We have further research in TB. We do it for HIV. And that's the link with the hackathon that we are discussing today. The link with the presentation that you will give later, I think, on the dengue hackathon is because we also have a drug in development that is active against the four different serotypes of the virus that causes dengue fever. And that's where we were thinking on how can we find data that predicts or find a model that predicts where the next outbreak is going to be. I'm almost done. I don't know whether I used up the 10 minutes, but no, but I want to keep to the standard. So I was visiting, we're doing in our company also a lot with external innovation. So we link and see where in this world, when you're looking for innovation in medical, in diseases, you cannot just discover it all on yourself by your own. You have to tap into the broad knowledge that is available. And so external innovation is part of our company that is pretty well developed, trying to get in touch and be a partner for those who are outside of a pharmaceutical company, who have innovative ideas, thinking, innovative drugs, and to see how can we collaborate, partner with them. And I was visiting in Singapore a building with all startups, and there was one little startup that the colleague that of Janssen that was working in Singapore told me, you have to visit that group. I said, what are they doing? They're data scientists. I thought, what? Why should I visit data scientists? Which was a big mistake and a lot of ignorance that I'm confessing here. And I changed my opinion, by the way, and this was a couple of years ago. So I went in and it was very interesting. A lot of young people, like all of you, very enthusiastic, very driven to find, based on their knowledge of data handling, analytics, data science, to find solutions. And they were discussing one of the projects they had done. They had won a grant by the government, and this was predicting in a competition with different companies, predicting what determines delays in the airline schedule. And of course, the first thing I thought, is it so difficult? Weather is probably the one. But of course, they understood that also. Everybody understood that. But there were many other factors that determined the delay in the flight schedules. And of course, aviation companies were very interested in getting a handle on that, how to predict where the delays are going to happen and how to mitigate and anticipate on that. But when they won the prize, or they won the grant, and when they were explaining, I thought, but if you can do that for flights of airplanes, why can't you do that for predicting an outbreak in dengue, or where the hotspots of HIV resistance are in the world? And for me, that was so logical. But of course, since I had a lot of talks with Serge and others, maybe not that easy. But I still think it should be feasible. Why are we interested in HIV drug resistance? I explained that we are very interested in HIV because we have developed a lot of new drugs in that field. We are working on a long-acting of one of the drugs. And we think that with an injection every month or even every 2 months, that probably we can control HIV replication. So 6 injections a year instead of taking pills every day. That is also a level of innovation, and we are doing that in our group. But what we're also always interested in is when somebody fails the first regimen. Currently, there is not that broad access to viral load testing, and viral load testing is the amount of viruses you can find in blood. And that is a good marker to see whether patients who are taking treatment for HIV are under control, because you want to get that level to undetectable, because we know if you do that, that all the nasty effects of the virus in the body is much better controlled. So that turns it into a chronic disease with a near to normal life expectancy. When a patient is failing on treatment, and that can be because patient is not very compliant or there is not continuous access to treatment, you have to take that treatment very regularly, of course. If that happens, and the pressure of the drug in the system of the patient, in the blood, or in the body, gets too low, then the virus escapes and has mutants that renders that treatment ineffective. And we need to know that early on. What is done now often in the developing world, there is no access to viral load testing. They test immune cells, CD4s. And when they start to decline again, that is a measure that treatment is failing, because that shouldn't happen under a patient that is well controlled. But the lag time between the virus that breaks out again and starts to replicate and the decline of those cells is too long. You've lost a lot of time already, and you need to you need to act earlier, intervene earlier with a new therapy to push down the virus again. In certain regions, they don't even do CD4 counts, and they wait until AIDS-defining illness symptoms occur again. But that is even farther away, and a lot of damage is already accumulated in the body. So we want to learn early on when a patient is failing to take appropriate measures, which could be if a patient fails because the patient isn't compliant, to make sure that they are compliant and to increase that compliance, or if there are signs of resistance occurring, to be able to change to a new treatment and therefore preserve the immune system and, in a longer term, the health of the patients. That's why we are interested. And knowing the viral load is extremely important, but also knowing where are these hotspots in the developing world, in resource-limited settings, where HIV resistance occurs. What we do in the developed world, we send a sample to the lab, and you can basically find any mutation in the lab. That is not accessible and not affordable in a resource-limited setting. So that is not the way forward. Maybe one day, I hope, that we have these sophisticated diagnostics too and lab tests, but currently that's not, I think, a feasible approach. What we need to know is to focus on those regions where there is known drug resistance, and therefore focus on, are patients infected with drug-resistant virus, or are they actually failing their regimen? That is important to know. And that is what I thought when sitting in Singapore, not difficult. We find all those parameters that correlate with higher incidence of resistance. This was a couple of years ago, it says. So we're still waiting. So that's why I think you can contribute to significantly to solve a piece of that puzzle. Maybe the entire puzzle. That would be fantastic. But I think the idea of bringing diverse minds together who are not hindered by any specific knowledge, to bring those together and to look at different angles to solve this equation, I look forward to the results of that. So I think I exceeded my 10 minutes.